but I don't know how.
All right.
There we go. We got the recording notice, so we're on.
We're on. We're recording at this point. All right. Well, let me reintroduce myself since it's now, I believe it's almost 6:30, or it should be 6:30. My name's Janet Lenachek. I am the chair of the Sierra Club Niagara group. And Sarah Schultz is our co-chair, which I don't think she can be with us tonight. John is part of our executive committee. You want to introduce yourself real quick, John?
Yeah, I've been on the executive committee for a number of years. I'm the one who talks trash in the group. I deal with the waste and recycling issues for our group. So if you have anything that deals with plastics or landfills or stuff like that, I'm the one to reach out. That would be reaching back out to you.
He's your guy. And John, I got a question for you after this. So remind me to ask you after the Zoom, because it has to do with plastics. Oh, Bob is on now. Hi, Bob.
Hi.
How are you?
Good. How are you?
Bob is also part of our XCOM with the Sierra Club Niagara group. And Bob, why don't you quickly introduce yourself? Let us know what you do with the group, so people who don't know you know.
Well, we work a lot on energy issues, renewable energy and energy efficiency. We've been supporting some legislation. We usually get involved in that. So we have a meeting every other week from the state. So it's a nice group. We get about 20 on the call. So nice to meet you all.
Great. And I'm on also part of the political committee. I chair the political committee. We try to elect people that care about the environment and are willing to support legislation at the county, state, and national level that aligns with the Sierra Club's goals. So, all right, without further ado, John, you want to introduce Courtney very briefly, or should we have?
Yes, I can do that. Good evening and welcome to tonight's environmental series presentation. When you hear the Sierra Club talking about going green, it's usually a good thing. But if you've heard of the annual harmful algal blooms in Lake Erie outside of Toledo, Ohio, you know that may not be a good thing when your water turns green. Our feature speaker tonight is Courtney Wigdahl-Perry. She's an associate professor at SUNY Fredonia. She is an aquatic ecologist interested in lake responses to natural and human-caused environmental changes on different spatial and temporal scales. She has a bachelor's and a master's degree from University of Wisconsin at La Crosse, and a doctorate in ecology and environmental science from the University of Maine. Last year, she was a SUNY Chancellor Award winner for excellence in teaching. And tonight, she will be talking about harmful algal blooms and her research in Chautauqua Lake on water quality and the blooms that are happening in both Chautauqua and other inland lakes in New York. And with that, I am going to pass this over to Courtney for her presentation. Thank you.
Wonderful. Thanks so much for the introduction, and thank you for the invitation. It's great to be here. I always enjoy a chance to get to talk to the community. I think that's such a huge part of what we need to do more of in science is share what we know in accessible ways and have opportunities for people to ask questions. So I'm really excited to get to chat with you all tonight. I'm going to go ahead and share my screen and nope. Okay, can you see my screen? The presentation? Okay, great. Excellent. I'm going to move this down, move this over. Okay, great. So There we go. Got two screens going, so I'm looking in two different places. Excellent. So I wanted to open up tonight by showing you a rather beautiful picture of our local site, one of my main research sites, Chautauqua Lake, because I'm going to be showing you some pretty ugly green chunky images soon. So I wanted to just start off by saying that Chautauqua Lake is a beautiful location. It's a really interesting lake to work on scientifically, and has a lot of value here in Chautauqua County. So I just wanted to preface the talk by starting there. So what we'll do tonight is walk through the basics of what are algae blooms, what does this look like in Lake Erie, and how does that compare and contrast to Chautauqua Lake as a different type of system. And we'll end up with what my lab and others are doing to understand this problem in our local system. So as far as, you know, this idea of going green, as John said, is usually a positive thing, right? Like, but in this case, it's not always great when it comes to water quality. I think it's really interesting with blooms to think about how the same issue manifests in different types of systems. If you look at these images from Lake Erie versus Chautauqua Lake, you can see a lot of similarities. But there's also some really interesting differences when we dig into the details of what happens with these bloom events. The group of organisms that are responsible for harmful algae blooms in freshwater systems are called cyanobacteria. So I'm going to talk about that a lot more. But they're really, they have a long evolutionary history. They've been on our planet a very long time. And so they're really have some interesting adaptations that make them really good at living and growing in adverse conditions and taking those opportunities to grow when they can. They're really like equal opportunity offenders, I would say. You know, they grow in just about anywhere they have a chance to do that. So before I get into the details of what this looks like, I do want to take a step back and talk about what are algae and what does this mean? And I do this because I get two questions when I'm out in the field. And one question is, how are the fish? Which I don't usually know that much about because I don't study them directly. But then I also get a question, when are you going to get rid of all the algae? So my personal mission in life is to make sure that people understand what algae are and why they're important to have in a healthy lake ecosystem. So algae is sort of an umbrella term, basically, for these microscopic aquatic plants. They serve as the foundation of our lake's food web. And what I mean by that is it's really the energy entry point into the system that fuels everything else that happens. And they do this through the process of photosynthesis, where they take energy from sunlight, use it to rearrange carbons together and build sugars. And that energy is then accessible to the rest of the food web. So we're going to talk in a moment about some of this variety that we see, but just like when you look out at the terrestrial environment, we see trees and shrubs and grasses and all sorts of different organisms that all fall into that sort of plant category. Algae are the same. There's a lot of variety within that. But at the base of the food web, at the heart of this, they really fuel the next level up in the food web, which are the zooplankton, the animal plankton, and then that's what feeds the fish. So for folks who are interested in how the fish are doing, I would say, well, look a little bit more closely and see what's happening below them, and you'll learn more about what kind of fish a system can support. I love this image. This was taken by one of my students. It's a microscopic image of a plankton sample, and I don't want to spend a lot of time here because there's a lot to see, but what I hope you take away from this is the variety and breadth of shapes and sizes and colors that are represented here in just this quick snapshot sample of algae from the open waters of Chautauqua Lake. There's a lot happening here, and there's a huge amount of diversity, which means that it's really complicated and really interesting and exciting from a scientific perspective to try to learn more about what drives each of these different members of this complex community. Just as a quick aside, there are a huge number of algae species that fall into this umbrella. The best estimates probably range around 45,000 different species, but it's probably way, way higher as we're learning more and more from genetic analysis that some things that we thought were the same are not the same. They're actually completely different species. So it sort of begs the question, like, why are there so many different species of algae? Why doesn't one species win? Why isn't just one thing present in the lake at any given time, right? Or even ever. And this question was so interesting and engaging to early scientists in the aquatic ecology field that they coined this term, the paradox of the plankton. So G. Evelyn Hutchinson back in 1961, who's considered sort of the father of modern aquatic ecology, said there should be a winner. Why isn't there a winner? And what it boils down to, it makes sense when you take that step back, but truly aquatic habitats are really dynamic. They're just constantly in motion. And so we have different kinds of habitats, streams, lakes, ponds, wetlands, oceans that are connected to each other too. They're not just operating in isolation, they're connected. And so there are changes within each system, but then also across this connected waterway perspective. So there's differences within, say, a lake, if you were looking at the surface waters versus the deep waters, or from the shoreline out to the open water. So we have spatial variability. And then also time variation. So what's happening in the summer is obviously very different than what we see in the wintertime, but still really important biologically. And then, of course, year-to-year variation can cause the lake to look completely different one year to the next. So all of this environmental variation and the physics and the chemistry means that we have a lot of opportunities for different competition to lead to high amounts of diversity. And when we generally look at conservation and think about what makes a healthy ecosystem, one of the common threads is typically that we have good diversity, right? We have a number of representatives from different groups that form a more resilient, more stable system, right? There's some redundancies, there's some backups. And so it sort of buffers a system from big disturbances, right? Big stressors. So it's actually really a great thing that we have all of these different present, but it does make it complicated to understand what's going on.
Transferring to
I don't know if somebody's needs to
Everybody
mute.
could mute, please.
Yeah, could everybody please mute on their Zoom screen? Usually it's in the bottom left. Thank you.
Thank you. Okay, so in general, we understand what controls algae growth, and a lot of these factors should look similar to what you might think of for your plants in your yard, right? In order to photosynthesize, you need light, you need the right temperatures. The key nutrients in aquatic systems are nitrogen and phosphorus. We're also often interested in sort of the physical environment and how water moves within the lake. And a lot of other chemistry can be really important, right? How acidic or basic it is, the salt concentrations, a variety of different chemical factors that might be part of that water chemistry setting. So these are sort of the general non-living or abiotic factors that control algae growth. In terms of the other biological factors that might be important, there can be competition between two different algae species or multiple algae species. And then you can also have grazing by the animal plankton, by the zooplankton, that can affect the growth rate of those populations that they're consuming. So we have a lot of different things. Like, we understand that all of these are important, but where it gets tricky is figuring out the exact combination of factors that might trigger something like a bloom event, right? So if you think about, one way to sort of connect to this is if you think about your landscaping in your yard, right? Like, we know that some things grow really well in more acidic soil, or some things need more light, or maybe it would be more shade tolerant. Algae are the same way, right? So we have a lot of those same sorts of things. Look, we know these are the big swings, but we don't know exactly always how these things sort of play out in the real world. These factors are also always in motion. They're always changing over the course of a season, right? So it's not just that light is important, but how much light we have will vary over the course of the year. And so I like this little cartoon that sort of walks us through from January through December and shows you the abundance of different key groups of phytoplankton or algae. And so what I want you to notice is that they sort of ebb and flow. And this is a simplified diagram, but this is the general basic pattern. In the spring, early spring season, late spring season into early summer, we have this group called the diatoms, which are another favorite group of mine in the algae community, that have this glass cell wall that are really cool. And I do a whole different sort of set of research with them that's really, really interesting, but I'm not going to talk about it much today. But diatoms really like sort of that low temperature, low light, lots of nutrients available in the extremes of the season. But they don't do so well. They don't compete as well as you get warmer in that middle season section. So the green algae come in and then they kind of fade out. And then the blue greens are the cyanobacteria do really well in this late summer season, right? In the heat of the summer, they like the warm temperatures, they can tolerate it pretty well. And they have some unique adaptive features that allow them to survive when other species can't during this time. But this is a normal sequence, right? So what I want to hammer home is that the cyanobacteria, which are a problem that we're going to talk about next, are normal. Like they're typically part of this healthy ecosystem, right? They just don't get kind of out of control until we throw the balance off. And so again, the take-home messages here are that total algae growth will vary over the course of the season. And then who is doing the growing, right, also changes. So sort of a quantity and quality issue. So algae and water quality, how do these things really connect? They're, like I said, again, normal, healthy part of lakes. But it's when we upset the balance and we get an overgrowth of algae that we start to have problems with water quality. And so when we have a HAB, a harmful algal bloom, this is typically driven by an overabundance of cyanobacteria, or those blue-green algae. This is true for freshwater systems. There's a different group of algae that grow in the marine system that form harmful blooms. Typically, those are what are called dinoflagellates that form the red tides, but that's a totally different issue. So I'm really only talking about the freshwater systems here and cyanobacteria. And so you might recognize some of the sort of scummy appearance when they accumulate near the shoreline. And they're again, fairly common across a lot of lakes in New York State and around the world. What happens when we have a HAB, are issues with the water transparency. So when algae grow really rapidly, we start to have less water clarity. It also affects aesthetics in terms of color and odor and appearance. And then these cyanobacteria can also produce toxins that are dangerous from a public health perspective. So they can affect our ability to use these resources as drinking water sources, and they can also affect recreation through beach closures because it's dangerous for people and animals to come in contact with the water at that point. From a non-sort of human perspective, the ecological health is negatively affected by HABs, because once all of that living material dies off, once the bloom is done, then bacteria process that and decompose it. And that decomposition process sucks up all the oxygen. And so when there's not enough oxygen around, then we have fish kills where large numbers of fish will die en masse because they can't survive those low oxygen conditions. Additionally, the food quality, like the nutritional quality of cyanobacteria, is typically not as good as other options on the algae buffet for the rest of the food web. And so we can have some long-standing issues with food web structure when we start changing what's happening at that base of the food web. So a little quick definition on how we define blooms. I'll tell you that this is different by state. So every state sort of has a different technical definition of what constitutes a harmful algal bloom event. Here in New York State, we have some pretty clear criteria, which is really nice from a scientific perspective. I will also just note that the DEC has really great educational materials and resources that are available on their website. So I absolutely encourage anybody who's around the water to make sure that you know what a bloom looks like and how to avoid it, and then how to report it. And so you can do this by going to the DEC HABs program guide and brochure and looking at some of those educational materials there. In New York State, when we have visible algae, we collect a sample for lab analysis. And all of that visible algae that's a very big category, because we have lots of different sorts of appearances, whether there's sort of small clumps, big streaks, different colors. It's not always bright green. Sometimes it's more teal. Sometimes it's red or brown or yellow or white, depending on the age of the bloom. So it's good to know sort of what it can look like in different places. Once we have that laboratory sample, in order to meet the criteria for a bloom, it needs to either have blue-green chlorophyll, the photosynthetic pigment that's unique to cyanobacteria. There's a special kind of chlorophyll. If that's at a certain concentration, if we look at the sample on a microscope and we see that it's cyanobacteria mostly, or if we measure one of their toxins they can produce at a certain level, then it classifies as a bloom. Okay. So there's different sorts of recreational levels, drinking water standards. There's blooms that are sometimes toxic and sometimes not. And so this is really helpful to have these sorts of criteria along with this. So New York State does a really good job of monitoring. In terms of what really specifically triggers a HAB to occur, The fine details are things that we're still actively studying. But in general, cyanobacteria, because they like that sort of late summer warm temperatures, they can really be set off when we have those high temps, but then also with excess nutrients that are coming in from the landscape through human activities. So things like surface runoff, lawn fertilizers, agriculture, sewers or septics that are not functioning properly, those sorts of things, right? All of that, anything that's happening up on the watershed can wash in, and those nutrients can then fuel the cyanobacteria that are already happily growing at that time. A few years ago, when the governor had a big harmful algal bloom initiative project, they did some assessment across New York State and identified 4 main risk factors that are associated with higher frequency of algae blooms. And those are high phosphorus concentrations, zebra mussels or quagga mussels, the drift-sanded mussels, the length of the lake, and then the orientation of the lake relative to the wind. And so these are 4 things that are tend to be associated with lakes that have high blooms. One of the things about cyanobacteria that's really interesting from their physiology is that they have a unique ability to create their own nitrogen. So we actually have a lot of nitrogen on our planet, but most of it isn't usable. Most of it is in the form of nitrogen gas, which is 2 molecules of nitrogen that are stuck together really, really tightly. So right now you are actually breathing in about 78% nitrogen gas, but it goes into your lungs and then comes back out. It doesn't interact with you at all. It's totally inert with your body. But cyanobacteria have evolved a special pathway that they can take that nitrogen gas molecule and break it apart, and then use that nitrogen, turn it into a more usable form. So they're basically they're accessing a totally different pool of nitrogen. This is really helpful in that late summer season when everything has been sucked up and there's not a lot of extra nutrients to go around. Cyanobacteria can bypass some of those issues. So especially if you have a lot of phosphorus, the blue-greens can take off, right? So it releases that limitation for them. They're also a problem, as I mentioned, because they do create different kinds of toxins. And some of them are neurotoxins, some of them are liver toxins. There's a whole array of different toxins that are produced under different settings. And honestly, it's very messy to understand the exact triggers for those, but they are a major public health risk. So that's why HABs are so problematic for us. And these are just some microscopic images of local algae samples that we've taken here. So that's the general overview of HABs and what we see in terms of what are algae, what are HABs, what causes them, how do we define it? And now I want to talk a little bit about HABs in Lake Erie as a way to contrast that with what I'm going to share with you about Chautauqua Lake. So when we look more broadly at Lake Erie, we see that most of the blooms that occur are here in the Western Basin. As John mentioned, it's pretty famous for the 2014 events in Toledo, where the bloom basically sat right at the level of the water intake and shut down the water supply for the entire city of Toledo for multiple days. It was a big problem. There are so many toxins present, you can't remove the toxins, right? You just have to wait for it to dissipate naturally. There's nothing you can do. You're just at the mercy of time. So this really was like the real impetus to sort of start to study this in more detail. And so there's been a huge investment in research, and we understand a lot more about what's happening with the Lake Erie blooms. Now, Lake Erie is huge, and there's a lot of different things that are happening. So up here where we're located in Fredonia, there's almost never blooms in this part of the system. All the nutrients that are coming in from these heavily agriculturally based basins are coming in here in the Maumee River and in Sandusky Bay. And all of those nutrients are sucked up by the algae here, right? And so there's not a lot left by the time the water gets out here. So we're good on this end as far as algae blooms go, but the Western Basin has a lot of problems. What I wanted to also point out is looking at the spatial variability in this and how interesting the spatial patterns are, even in a huge lake like Lake Erie. It's not uniform. So it's interesting to see these satellite images. For scale, I want to point out how big Lake Erie is in comparison to Chautauqua Lake, where I mostly work, which is here, this little bit. And in comparison, Chautauqua Lake is a 13,000 acre lake, which is not small by any standards, right? It's a very large inland lake. but compared to Lake Erie is tiny, right? There's a very large spatial differential here. So a lot of different kinds of lake ecosystems that are all experiencing these blooms in slightly different ways. So kind of continuing on this trend of looking at satellite data, this is a series of satellite images looking at bloom intensity. So the red colors mean high bloom intensity, the purples and blues mean low intensity. And this is looking back since 2002. And I think this is such an interesting look at the Western Basin, and this is like peak bloom for each year, is what we're seeing here. And what I want to just point out is how much variation we get year to year, right? Some years like 2002 and 2005 have very little bloom activity. And then you could look at other years like 2011 or 2015, and it's really, really bad. And it's widespread. And some years you have pockets where there's a lot of bloom activity in one space, and the next year there's nothing. So for me, when I see this, I'm really fascinated by the different patterns, right? And it makes me want to know why these things are happening in different ways across the system, right? I want to know why they do that. Why are they here and not there this year and then back again the next? So it's an interesting question and complicated when we start thinking about the physics of how stuff moves around in a really big system. So just a little flavor on Lake Erie for harmful algal blooms, but HABs are definitely not unique to large lakes. And though the Great Lakes get a lot of press and a lot of focus, overall, most of the lakes on our planet are very, very small. So we have just a handful of lakes around the world that fall into this Great Lakes category, right? Of the 304 estimated million lakes worldwide, like 303 million of those are, or more, are all small in surface area. And when I say small, I mean less than 1 kilometer squared. And so 1 kilometer squared is about 250 acres. So my frame of reference for that is Bear Lake, which if you've been in Chautauqua County and been to Bear Lake is about 112 acres. So most lakes are twice the size of Bear Lake, which is a small kettle lake, or smaller, right? So HABs do occur in these smaller systems because often they are also shallower, which means that they're warmer. And they're often situated in locations where we have high human impact, which leads to more and more nutrients coming into the systems. So small lakes are just as susceptible to bloom issues. And these affect their ecosystem health and the services that we get from these lakes. Again, the public health issues, recreation, economic impacts, and then also using these smaller systems for drinking water becomes much more complicated with bloom activity. So shifting to a slightly smaller, but still pretty big inland lake, I want to shift us to Chautauqua Lake here in Western New York. That is one of my primary study sites. So as I mentioned, Chautauqua Lake is a 13,000 acre lake, about 50 square kilometers. It's what I would call a dual basin lake. It has a slightly deeper north basin where there's a maximum depth of 76 feet in this deep hole. And then there's the south basin, which is more like a max depth of, I think, 16 or 17 feet, not very deep. It flows from the north out to the south, generally speaking. The south tends to be more productive. So there's more algae blooms that happen in the south basin, but they do also happen up here in the north. And so Chautauqua Lake has frequent persistent algae blooms. So there's a lot of interest in trying to figure out why they're happening and understanding more about this action in the lake. Chautauqua Lake is a huge economic driver in Chautauqua County. It's a drinking water source for a few communities around the system. It's a major recreation tourist destination. And there are also world-class fisheries at Chautauqua that people come from all over to fish for muskie and bass and walleye. And all of these things can be negatively affected by these frequent and persistent blooms. So I always value perspective, right? I think time perspectives are really important and really powerful. So it's very helpful to have, we're really fortunate at Chautauqua to have been one of the first members of the New York State Volunteer Monitoring Program called CSLAP, the Citizen Statewide Lake Assessment Program. It initiated in 86 or 87, depending on which metric you're looking at, and has been consistently monitored except for maybe 1 year in the mix there. And this is biweekly sampling throughout the summer season. So we have some really excellent data from the North Basin as well as the South Basin to track longer term records of environmental change. So there are three things we typically look at related to lake water quality, sort of three main things that connect to algae blooms. And the first one is clarity. And so water transparency is a really important thing for us to track for a number of reasons. And we've done this historically by using a super complicated tool called a Secchi disc. It's actually just a painted black and white disc that you lower over the side of the boat and watch when it disappears. So it's a simple tool, it's easy to train people on, and it's incredibly useful because it's a standard for water clarity through time. And so this gives us a sense of, okay, if the algae growth in the lake is low, if there's not a lot of activity, the water is clear, so we'll have a deeper Secchi reading. And if we have a lot of algae activity, the water is not clear, it's more turbid, and so we have a very shallow Secchi reading. So what you can see here is that it's relatively stable, although we had some years that are much clearer than others, and some that are more shallow, right? So the water was not so clear. It allows us to sort of assess the state of the system. And so we use terms eutrophic, mesotrophic, and oligotrophic to sort of indicate how productive or how much algae growth is happening in the system. So eutrophic means highly productive or sort of on the green side of things. Meso is in the middle, and oligotrophic is low productivity. So sort of clear, deep blue lakes, right? So Chautauqua is usually hovering around the mesotrophic or eutrophic category, which is what we see here in the Secchi reading, and that's true for other metrics as well. Another thing we measure is the algae biomass itself through looking at chlorophyll A, which is the main photosynthetic pigment that all the algae share, right? They all have it. And so if you look at the amount of chlorophyll that's on average for the summer, you'll see that there's typically we're well into the eutrophic category, but there's a lot of variation, right? There's a lot of some years are really, really high, and then it drops off the next year. So there's a lot of, again, a lot of variety, a lot of variation in terms of how much it changes from one year to the next. Overall, at least for the last 35 years, it's been relatively stable. Like we have some ups and downs, but it's not like skyrocketing in this case, right? So that's good. The last thing that we often look at is the phosphorus concentration. Since this is one of the nutrients that controls algae growth, we want to make sure we're tracking how much phosphorus is in the lake. And so again, looking at this category, we're sort of just into the eutrophic zone, looking at the surface waters as well as the deep water phosphorus. This is trending up slightly, although it's not a really strong trend based on the last few years as well. So I think this is really good. What it shows for us here is that in general, the lake is productive, but generally stable. There's some good reasons why this might not tell the whole story, but that's what we're looking to find out, right? That's what we're working on from a research perspective. The CSLAP data dashboard, if you want to learn more about this or other systems that participate in CSLAP, there's a new data dashboard that you can go to here. You can just Google CSLAP data dashboard and it'll pop up. And so it's really accessible, and it's an amazing program, and a lot of work goes into this. So we're very lucky to have this data set. Very cool. In terms of some additional work that's been done more recently, looking at the blooms specifically, again, we have frequent persistent HABs. In 2021, there were 49 HAB reports that were called in to the state from different sources. And they categorize these by open water toxin concentrations and shoreline toxin concentrations. The good news is that with open water blooms, we tend to not see really high toxin concentrations, at least in 2021. The shoreline is a different story. We do tend to see more toxins that we measure, right? Higher concentrations of toxins in those shoreline blooms. So from a spatial perspective, it's also interesting that we're seeing not just spatial differences in the algae makeup, right? Who's living where in the lake, but also in the toxin concentrations that we're seeing. So a lot of interesting data, but what typically happens in science is this creates more questions than answers. So that's what we're after here. And I wanted to show you two quick videos. These were from a bloom. I told you I was going to show you green and chunky videos, right? And so these are two videos that I took from a bloom in October 21. So this is a pretty late bloom at Chautauqua Lake. And this is this happened in the South Basin, and it covered basically the entirety of the South Basin. Almost 5,000 to 6,000 acres of lake had this very calm day, had all of this surface scum that was present. And this is a slightly different view that really captures that bright green color. I like the first video because it shows you the scale of everything, but this one gives you that really close look at that surface scum and how thick it is. So it's a huge amount of algae that was living in the lake at that time, and they came up very fast. So we have this really valuable local resource near the university, and I'm really interested in trying to understand more about it. So my research lab has been very active on Chautauqua Lake since I arrived at Fredonia in 2014. I kind of center my questions, and we do a lot of things. We're a very busy lab, but we center the research questions around how do lakes respond to different types of environmental change. And we use this as a springboard to really focus on, in this case, understanding blooms at Chautauqua Lake in space and time. I'll also note that this is, I'm primarily actually teaching faculty, so I do a lot of active research, but most of my job is actually teaching and working with undergraduates and a handful of master students. So the work that I'm going to present today is primarily student-driven and collected by some very intrepid early career scientists who are doing a really excellent job. So if you see us out on our little raft at Chautauqua Lake, please say hi. We're always happy to share what we're up to. So as I said, we're a busy lab. We have a lot going on. I'm just going to give you 3 little blips in the interest of time. And I can be happy to answer more questions about things later. But I just wanted to highlight a few things that we're doing. And then I'll also mention briefly what my colleagues are also doing. We're very fortunate to have a number of additional researchers working at Chautauqua. So one of my graduate students, Casey Crandall, is working on this question of what's happening with temperature in Chautauqua Lake. And we're really interested in the vertical temperature patterns from the surface to the bottom of the lake, because in lakes around here, we end up with these 2 layers in a process called stratification, where we have a warm surface layer that stays separate from a cool deep layer, and they stay separated pretty stably for basically the entire summer. And this separation, right, this no mixing between these layers means that there are really different environments for chemistry in the surface versus the deep. So the nutrients are different, the pH, the oxygen concentrations, the physics of what's happening in the surface versus the deep waters are sort of these 2 whole like separations in the lake that are occurring simultaneously vertically. So it's very interesting. And we've been exploring this in a lot more detail since 2019. And this just shows you a heat, we call it a heat map, where we look at temperature through time vertically in the water column. And you can see here this nice, warm, stable upper layer compared to this cool, deep layer, and how they stay really, really distinct for the season. So this really led us to some interesting questions, and we're attacking the problem with a little bit more technology now, using temperature loggers that we put out in the field, and we leave them there on temperatures on strings, right, on ropes that go from the bottom of the lake up closer to the surface. And these sensors read independently without us doing any additional work then in 5-minute intervals. And we space them out about 1/2 to 1 meter in space, so about the length of your arm apart. And so we get this nice array of data that's really high resolution. And so we've learned a ton about what's been happening in the North Basin and then in the South Basin. And our goal this year is to deploy 5 lines simultaneously across the lake to see how they're interconnecting, right? Like, how are these temperature patterns affecting each other in the North and the South Basin? So we'll have 5 lines spread throughout the lake that are logging 5-minute intervals the entire season. So that's very exciting. So we've got 1 line out, and hopefully we'll get the rest of them out next week. My other graduate student, Cameron Finch, is doing some really cool work on road salt, which is new for our lab and actually fairly new for Chautauqua. We haven't asked much about what effect road salt might have on our aquatic ecosystems here in Chautauqua County. In general, the US uses on average almost 25 million tons of road salt every year on our roadways. And it makes sense because we want to be safe driving, right? But unfortunately, that salt does not stay where we put it. And then we have to keep going back and applying more. So the salt doesn't just go away. It typically runs off into streams and then gets flushed into lakes. And this is a nice little figure just demonstrating a long-term record of a forested stream in New Hampshire, here along the bottom, that is not near a highway. And then looking at this other stream that is right next to the highway, and you can see the chloride concentration going steadily up by quite a lot compared to that baseline value. So we know that the salt is moving, and we know that it's increasing in concentration in our waters, but we don't know exactly what that means for the biota. The EPA set a guideline for natural waters back in 1988 at 230 milligrams per liter of sodium chloride. But it's complicated when we start talking about storms and flushing and pulses versus average. It's a little bit messier than that. And in particular, guideline was set based on zooplankton, animal plankton that feed on the algae. And so there's a lot of interest in more broadly in the scientific community and understanding, is this guideline actually going to be effective for us, or do we need to sort of revamp what we view as safe? And so Dr. Shelley Arnott at Queen's University in Canada is leading a global effort to do salt tolerance experiments with this little zooplankton called Daphnia. And so people around the world are participating in this experiment, and us included, where we collect local zooplankton from our systems, right? Not laboratory strains that the EPA use that have lived in a lab for, 50 years, but things that are directly from the environment, and bringing them back to the lab, growing them up here, and then performing salt tolerance experiments to see how much can they actually handle and how does it affect their survival rate. So this is a new project for our lab, which is going to be really exciting. We live in a place in Western New York that requires a lot of road salt. But there might be some ways that we can adjust the way we approach this in order to still be safe on the roadways, maybe save the town some money, and then keep our waters safe and healthy. The last little vignette I'll mention is that I've said a few times how the spatial dynamics of blooms are really interesting, right? How things vary from shoreline into the open waters, and how there's just a lot of variability. And so this last little piece I'll talk about really came from this idea of, we want to know more about bloom dynamics. Where do they come from? How do they grow, expand, contract, move, dissipate? Our traditional sampling techniques, where we go out on the boat, collect a sample from one or two locations and go back to the lab, doesn't afford us the opportunity to do a lot of spatial perspectives. So for larger systems, we've been using this sort of satellite imagery with remote sensing. So remote sensing is all about getting information from an object without actually touching it. So this is what we see satellite images. This is sort of the classic example. Now, this works really well. If you remember what I said, so that size differential, right? This works great on Lake Erie. But for Chautauqua Lake, it's not as effective because it's a much, much smaller system. And if you remember, Chautauqua Lake is pretty big compared to the size of most of the other systems that we have on our planet. So satellite remote sensing approaches don't always translate well to smaller lakes because of resolution, flight timing, cloud cover. We want to look more closely at the shoreline, and we can't do that when the lake is that small compared to the satellite cover. Additionally, though, there's been some really cool work done on algorithms and models for large systems. Honestly, there's different species of algae that we find in Lake Erie, right, that they're building these models on, compared to what we see in lakes like Chautauqua, right, or other smaller inland systems. There's just different communities, right, different types of species present, different colonies, different shapes and sizes and pigments. And so we really want to use remote sensing, but we need to do a better job of using a different tool that lets us get that closer look with as much detail as possible. So this was where this drones and hyperspectral imaging project was really born. We're using drone flights. We can fly closer to the lake with a hyperspectral imaging camera, or HSI camera. Corning Incorporated was generous in supporting this project by donating funding and cameras for us to work with. What's unique about this HSI is that it gives us a huge amount of information per pixel, right, compared to what you might get from sort of a standard camera on your phone, right? That has 3 bands, red, green, and blue. HSI has 155 bands compared to the 3. So we get a lot more spectral data, which should let us actually separate out those cyanobacteria that we're really interested in from all the other algae groups, which is exciting. But this has not been done before for inland lakes, especially not small ones. So we are initiating a sampling effort with a lot of ground truthing. And so we have underneath the drone flights, we're doing up to like 20 samples in a very small space to get all these different algae metrics. And that way we can start to build these data sets together to understand what is the camera really detecting, and how can we apply this in the future. So we've been working on this for a couple of years, and we've just got another new grant from the Air Force Research Lab. But this is a big collaboration with a lot of different folks across the New York State. So it's been a great team, and it's a really exciting project. This is what it looks like compared to like Landsat pixel size versus the drone flight, just so you can kind of see how different that is. And what I love about this drone flight is that you can see the blooms in between the docks, and you don't get any of that resolution with a Landsat. Now there's pros and cons to all of these methods. None of these are perfect. They're all challenging, and they come with their own strengths and weaknesses, but it's very cool to be able to take a really close look at these key shoreline areas. I'll just note that this was done during COVID, which was a particularly difficult time. So I'm really impressed with the Binghamton students that did this work. So we have a few ground truthing samples. We do a lot more now, but they were operating under some pretty difficult circumstances. So this is an impressive amount of work that they did. Briefly, we have a lot of other research partners at Chautauqua Lake, which is very exciting. The Jefferson Project is based at Lake George in the Adirondacks, and that's a partnership between IBM, Rensselaer Polytechnic Institute, and the Lake George Protection Alliance. So we have our industry leader in computing, an excellent academic partner, and then a community engagement connection. So it's a really cool project. And they've been at Lake George since 2013, and now we're establishing a Chautauqua Lake outpost. And so we have now 2 vertical profiler buoys that take continuous water chemistry and weather station information. They're also, so there's one in the North Basin and one in the South. They're also doing bi-weekly open water sampling, and some benthic or bottom algae monitoring. And then all this fuels into computer models. So we can eventually get to the point where we can forecast what's going to happen. We have additional collaborators at Bowling Green State University that have helped with a number of things over the last few years. But this year we're doing some additional stream sampling from 3 Chautauqua tributaries to look at the water chemistry. And so we have 3 locations this year, and at each stream we're doing upstream sampling. So we start at the mouth and we go up into the watershed, and we're basically trying to see what's happening with the nutrients over the course of that run, so we can understand where we might be getting nutrients from, what that looks like as it goes through the system, etc. So there are plenty of other folks who have done things. These are just 2 of the teams that have been here more consistently over the last few years. So lots of other folks that have come and hopefully will come again. So in summary, algae are a normal, healthy part of lake ecosystems. This really diverse base of the food web is an important part of our lake life, and we want it to be there. But when we throw things out of whack through human interactions and activities, we end up with some blooms that can cause negative water quality implications. These blooms happen not just at Lake Erie, but also at Chautauqua and many other smaller systems. And a really active research program that attacks the problem from all sides is our best tool in understanding these issues going forward. So I'm happy to share this as well, but there are a number of resources if you'd like to learn more from the state. So and then the CSLAP reports from NYSFOLA. So please feel free to ask me if you'd like any additional information on this. Many people to thank. I have a huge number of really talented current and past students, many community partners and research colleagues, many different funding sources that have helped me since I've gotten here at Fredonia. And additionally, I'd like to thank our summer research endowments that help support our students over the summer. And so with that, I'll take any questions.
I was wondering, after the research, have you have local homeowners who have lawns along Chautauqua and some of the farmers who use fertilizers been contacted and maybe
Yeah, there are a
asked
number
to
of.
train or given options as to what they can use instead of
Mhm.
chemical fertilizers?
Yeah, there's a lot of education. The Chautauqua Watershed Conservancy has always had great materials out there to try to help people understand why lawns and fertilizing your lawns all the time can be difficult. They do a lot of training education for shoreline buffers and encourage native plants and rain gardens. So there's a lot of opportunity. But you know, we have a culture that really values lawns. And so it's sometimes hard to change perspective on what that looks like or what you want to have your property be. But there's a lot of opportunity, and a lot of groups will come around and help you plan things. So there's certainly many efforts in that area, and there've been some great changes for many of the locations. And so doing more of that would be great. The Soil and Water Conservation District has worked extensively with farmers and helps to connect them with different agricultural grant opportunities to try to support financially the improvements to the farms to reduce runoff and to minimize loss. You know, like a lot of agriculture can fuel these nutrients coming in, but it's important to remember too that farmers don't want the nutrients to leave their land, they want it to stay. So finding different ways to help them minimize loss, right, and increase their yield is really the goal, because it'll help them and it'll help the lake. So yeah, there's a huge amount of community engagement around these things. And I think one of the best messages is to realize that just because if you can't see the lake, it doesn't mean you do not have an effect on the lake, right? Water runs down. And just because you don't see where it ends up doesn't mean that your actions don't have an impact. So it's just an important perspective to gain. But a lot of people are working very hard on it, which is great.
Courtney, I have a question.
Sure.
I'm just wondering about septic systems and human sewage.
Yes.
Does that, I mean, I don't know much about Chautauqua Lake, but I know that it does overflows and things like that can certainly affect nutrient levels.
Yep.
Isn't there phosphorus and nitrogen in human sewage? And I'm wondering, on Chautauqua Lake, are there a lot of septic systems and things like that that may be adding nutrients as well?
Yeah, and it's one of the nutrient sources that we actually have a lot of control over. It's not cheap to replace a septic system, but it is something that we can actively control. So it is one of those pockets of nutrients that is really good for us to pay really close attention to. And so the county health department works on septic system surveys. So a lot of folks have really made some nice improvements. One of the biggest things that I'm really excited to see, though, is that the extension of the sewer district, the South and Center Sewer Extension, that's going in around the other side of the lake along the western shore. And so that'll actually connect some of these locations that didn't have an option for sewer hookups, and will hopefully help centralize our waste processing and avoid the expense of maintaining your own septic system. And also, you can't have a septic system that's functional like 50 feet from the lake, right? Like there's not enough leach field. This is not going to work. And it's just the physics, right? There's not much you can do. So the more we can try to avoid failing septics or failing sewer lines, things like that, the better off we'll be, right? So absolutely trying to minimize those sorts of issues is the way to go. So it's very exciting to have the infrastructure investment that's going all the way around the lake now. So I believe the first phase is going in now, and then the second phase of going in next year or the year after. So my sense is that they anticipate it'll be done maybe by 2026. I don't know that for sure. I have to look back at the county information, but yeah, so we're working on that now. It's not there yet, but that should help.
Good.
Yeah.
Any other questions for Dr. Courtney? I'll call her Dr. Courtney.
Yeah, I had a question. Is there kind of a one-to-one correspondence in the the more likelihood of having a harmful algal bloom versus the depth of the water body, or is that just coincidental?
Well, it's a great question. One of the things that happens is shallower lakes are typically warmer. And so they're the cyanobacteria tolerate that heat pretty well. And so that's kind of their natural like wheelhouse, right? Is they like those August temperatures, they do just fine. And so they're more tolerant of some of those high temps than some other species, some other groups might be. And so shallower lakes are often a little bit warmer. But it's not just that if you can have deep lakes that are very productive as well. So it's a number of factors that come into play, right? How much nutrients are being flushed in, how there are what we would call low productivity, like clear blue lakes that do get blooms, and that's happening more and more. We're recognizing it more and more. So one example of that is Otsego Lake last year. They shut down Glimmerglass State Park, which was like the first time that had ever happened. And Otsego is considered very oligotrophic, clear blue, deep, has never had blooms. And there was a big enough one that they shut down the park. And there are cases now where even what we would call low productivity lakes are having bloom events. And it's because of some of these physics of how those layers are, we think, some cases at least, I should say. The surface waters have different chemistry than the deep waters. And if there's a break in that, there's often higher nutrients down below, and that can bleb up and basically fuel blooms in the surface waters if there's a disruption in those physics, if those layers are not separated anymore. So we're learning more about this through all these high frequency data sets. A lot of it is, again, tied back to temperature, but you can have shallow lakes that don't have much algae growing if there's not a lot of nutrients coming in, or if there's enough or if there are other things that are growing that are taking it up instead. So if like there's more vegetation in a really shallow lake, that might suck up enough of the nutrients that you don't really have an algae problem. Depends. Yeah, it's an interesting question.
All right, Judy, can you unmute yourself?
Yeah, I have a couple questions and a couple comments. I wanted to know with the climate crisis that we're having right now, if that's going to increase this when you're saying it's the warm water. Have you seen any correlation between that?
Yeah, it's a huge concern. Not just because of temperature, but because of, so temperature is one aspect of it, but also our precipitation patterns are changing. And so in some places, we're still getting the same amount of total rainfall, but the delivery is very different. So for example, we might get 100 millimeters of rainfall in a given year. But what used to happen is we would have a lot of days of really sort of gentle rain, right? And it would soak in. Now what we're getting is drought, big rainstorm, drought, big rainstorm. If you've ever forgotten to water a plant and then had to dump water on it, it's like practically hydrophobic, right? Like the water just like sheets off of it. Is a very different process when we're talking about transport from the watershed. So these pulses of nutrients come in, or a lot more stuff gets transported in these big storm events. So storm frequency and intensity is also a big part of this question mark. And that's much harder to predict. Precipitation is much messier when we look at climate models. It's just not as clean cut, right? So we're learning more, but it's tricky. So in terms of climate change, unfortunately, when we look ahead, the things that, contribute to cyanobacteria blooms are going to continue to get worse, looking at increasing temperatures, looking at longer growing seasons that allows them to stay later into the fall. Lack of ice cover in the winter means an earlier spring. So if the lakes don't ice over, we end up with a much earlier spring, and that can change nutrient dynamics and who lives there year round. So climate change is such a multifaceted issue, but in general, we anticipate that the current changes we're seeing are going to make it more likely to have bloom events. So it's one of those things where Chautauqua Lake right now, by the measures that we have, has been relatively stable, which is good news. Those samples might not tell the whole story though. Like it's a great data set, but the amount of work that you create for yourself every time you go out and take a scoop of water is like, I tell my students, okay, you can go sample, but for every bottle you're collecting, we're going out today, but you've just created 3 weeks of work for yourself, right? So it's not as simple as, oh, we just need to sample more in more places and this will be great. It's like, no, that's not physically possible. So there are some sensors that allow us to shortcut some of that workflow. We get less information more broadly, but we get better information on like one or two things, right? And so sensor data can help us understand some of these things, or some of these spatial perspectives, like the remote sensing approaches, help tell us a little bit more about what's happening in other parts of the lake, not just the ones where we go to this site and that site, so there could be more that's happening that we're not catching in the monitoring program that currently exists. And it's no fault of the program, it's just the nature of how that operates. But that's where it gets really exciting to bring in these other tools, right? With multiple research teams that are all attacking from a slightly different approach. And then we collaborate, right? So we can bring all these ideas together. So, looking forward right now, we have a lot of blooms, but I don't think that they're necessarily better or worse. And anecdotally, every time you talk to somebody around Chautauqua Lake, it's a different story. It's as bad as it's never been this bad, or it's the best it's ever been. It's so fascinating because the perception of water quality is highly variable, right? Which is why the data really help us sort of, and it's not that those things aren't true, it's just that what somebody experienced maybe in the South Basin in the 50s might be different than what somebody experienced in the North Basin in the 70s, right? It could just be from one bay to the next, even in one section of the lake could be totally different. And so I think this era of big data is going to be really helpful in us figuring out what we have and what are the patterns. So I am really, in terms of like future issues, the harmful algal blooms are not going to get better without a lot of work. And I think the more work we can do to limit nutrient inflow and to understand these things better, the more likely we're going to avoid bigger problems down the road.
Yeah. What would be your recommendations legislatively? I mean, what should we be pushing for?
Yeah, I mean, I think the standards are really to reduce nutrient inputs into the lake. That's the biggest thing that we have control over. Certainly the climate change is the biggest, baddest background. But we have, in terms of a community, have the least ability to affect that change directly at the lake, right? We can all operate together on a bigger platform. But that trajectory is set. That's going to be moving in that direction for a while. Even if we all stopped driving cars tomorrow, we are going to be experiencing climate warming for a time to come, right? But locally, the biggest things are really working to reduce any transport to the lake. Anything we can do to slow water down is good, because all of the plants and vegetation, bacteria, all the other living things on the watershed are pretty good at soaking that material up, right? And reducing nutrient input before it gets to the lake. The more you can slow water down, the more you can reduce direct runoff, using smart fertilizers, being careful about your timing of when you're applying these things, chemical treatments on your lawns. Like you can still have a beautiful lawn, right? Just don't apply it year round every week, right? You don't need to, it's not going anywhere. Do it like in the spring and then once in the late fall, right? Like those sorts of, you can still have your lawn. You don't have to give up everything. But that can help, right? Like every little bit is going to help us reduce the amount of nutrients.
I have a sign in my front yard, grow food, not a lawn. And that's where I grow it. Are you working with our legislation then in this county for Chautauqua Lake? Because I know that that's a really big issue.
Yeah.
Lisa probably knows a lot more about that than me, but they're always talking about what they need to do. And so do they call on you and ask you for?
Some of them do. I'm always happy to be a resource. I put my name out there a lot. I'm at community events all the time. I do these things all the time. Good. So I have worked with several of the legislators, quite closely. It varies depending on who it is. But some folks are really big advocates for lake research and support. And I think that's been really nice to work with different folks. So I'm always happy to answer questions and be a resource.
Lisa has a question, I think. Lisa, unmute yourself. There you go.
I just wondered about your opinion or your findings about the effect of the herbicides they were using in the lake.
Mm-hmm. Yeah, it's
very controversial.
Yeah, no, so it's such an interesting topic, right? I really come at it from the science side, right? Like, in my perspective, and I will tell you, I'm not a lake management expert. And that's because lake management is way more than the science. It's so much more about people. And I like people. But I really like the science. So this is really my playground. So what I look at in terms of the scientific information that we have on lake management is that we have big problems in this world. We need a lot of tools in our toolbox to pull from. And we need to be careful about how we use things and intentional and reflective. Good lake management should be adaptive. We shouldn't take things off the table ever, right? We should be looking at things and carefully weighing pros and cons, right? Everything has a trade-off. Doing nothing to manage the lake has negative consequences, right? Because we have created this problem, we have created this beast, and we could do nothing. We could say we're just going to leave it alone, and that would be bad, right? Because we've created the problem. So even doing nothing has negative consequences. So where I come at it from whatever activity that might be is to really think carefully about what are the pros, what are the cons, short-term, long-term, what are the possibilities, right? And then help the community understand those components and weigh that, because the decision on how you handle these issues is beyond just the science. I want the science to be the foundation for this so that you have all the information, but then your community values come into play with lake management, right? And your goals and your targets, which are very much community driven by people. So I think that when you're looking at invasive species and herbicides, I think it is absolutely a tool in the toolbox. It can be very effective to get something done quickly. You know, it is a chemical you're putting in the water, so there are things you want to pay attention to. We have a big fishing community that's really, really a major economic driver, but also so is recreation and businesses and people that are suffering because people don't want to come stay there because there's weeds and they can't get rid of the weeds. But you know, so I think there's so much push-pull, and what I really hope about that issue is that people can think about the real scientific information that we know. And I think that you know, that's where I come from it. Like, I think that the plants are a separate issue from the algae, but it's in this living dynamic system, right? Sometimes algae and plants compete with each other. So like having more plants is actually good because they suck up the nutrients and they reduce the algae. Other times they're mutualistic though. So if you get rid of, if you have more plants, you have more algae, right? They're different in different systems. The relationships are complicated.
Well, if you go to the annual lake conference, it's coming up again in June.
Yep, I'm speaking again.
Well, a couple of years ago, there was someone, I think from the Finger Lakes region, who was a management scientist, who said that the herbicides just threw it too far out of balance.
It is a concern.
And he had a lot of good reasons, you know, for that were database, you know, evidence-driven reasons. And I kind of, you know, went to him, you know, because that seems, I don't see how, you know, poisons can be beneficial to anything. You know, it seems like they could kill the fish. And then some of the, I can't remember the name, a couple people here who are, you know, involved with that felt that that the herbicides could add to the proliferation of algal blooms.
It's one of these really complicated things, right? So, I mean, I think we can all agree that ideally nobody would want to have to harvest or add herbicides. Ideally, we wouldn't have to worry about this stuff.
They're harvesting, they have that big machine, they go out and
but
pull out.
all of the management techniques have a con, right? They all have a pro and they all have a con. And, you know, the chemistry is super interesting. And I will say I am not incredibly well versed in the chemistry of like Procelicor, which is used for the milfoil treatment. Milfoil is an invasive species. It's bad for boats. It is not native. It's been here a long time. It's aesthetically problematic, but it also outcompetes native plants. So one of the things that's really interesting about Procelicor, which targets the milfoil, is it's very specific. There can be probably in the real world some collateral damage, but it's very specific to the plants. It's very specific to milfoil. So the chemistry has gotten really targeted, which is interesting. So there are more general, like broadleaf, like killing everything kind of situation, but the chemistry has gotten really interesting where some of these things are quite targeted. It doesn't mean that we fully understand what happens in the real world. It doesn't mean that we necessarily know all of the ramifications, But where I would come from is looking at the science of it. It is regulated. The applications are very tightly controlled. The DEC covers all the permitting, and they come out and check the area to see if it's warranted, right? So the municipalities apply for the permits, and then the DEC comes out to check before they're allowed to apply. So it is not like a willy-nilly, like, I'm gonna go just nuke my lawn or whatever, right? Like it is a tightly controlled process. But again, it's a tool in the toolbox. So all our alternatives are to go through and you know, you're investing a lot of money no matter what, right? But you have pros and cons for all of these things. So I would always like come at things more cautiously and say there is no free lunch here, y'all. Like there's always going to be something that we have to weigh and measure. And I you know, nobody's going to like the idea of applying things or spending money on things you don't need to do, right? But we again have, through human activity, amplified these problems. And then we want to use it, right? Like the lake wouldn't care if the weeds grew there, like if nobody wanted to go swimming and boating. So our human interests and our value in the system affects what we choose, right? And what we're willing to sort of take a risk on, what we're willing to pay for, and how we approach those problems. I would love to see more collaboration and less like this is a hard no all the time versus this. Like I would really like to see more ways of, okay, this lake is changing every year, is so different. Here's my analogy. If you were to go to the doctor because you had a heart attack, right? And the doctor said, okay, for the next 40 years, you're going to take this same dosage of this same pill every day. We're never going to change it. We're never going to check how it's doing. It is just, this is what we're going to do. You would never stand for that, right? You would be like, no way, I'm changing. I am doing something different in my life. My lifestyle is evolving. I have different considerations. This medication stopped working. So maybe we need to look at something else, or maybe my dosage needs to be changed, or maybe it's doing fine and maybe I don't need it anymore. But that's sort of the mentality of adaptive management, right? Like think like, let's look critically, let's get data, and let's have good conversations about evidence, and then let's do our best to collaborate. Because what works one year might not be needed the next year, or maybe it will really be needed. And I think if we limit ourselves to only one pathway towards helping the lake, I think we are not necessarily putting the lake's best interest at heart, which is what I would really love to help have that be more part of the conversation. So it's really difficult. There is no clean answer. And an opinion doesn't matter to me, right? It doesn't matter to the bigger picture. What I want to do is help look at the science and make sure that we're really not getting caught up in, and he said, she said, they said, like, let's really like solution, let's problem solve, right? Let's work together and keep everything on the table. We got big problems. We cannot afford to not have conversations about it.
I just live up the hill from Lake Chautauqua, and I had to replace the sewer pipes in my yard. I don't have a septic system, and I have, because they are really, really old and they are leaking. So and there's a lot going on here that I'm aware of, you know, and I read all the material and listen to the presentations and stuff. So, you know, but that makes me not want to go in the lake. I enjoy it from the shore.
I was going to say, Courtney, I love your analogy of thinking of the lake the way it is now as sort of an ill patient. Right. You know, like, how are we going to approach this to help improve the health of this lake? Let's look at all of our options. What do we want to try? What could we afford to try? And if it's not working, try something else, you know, go back to the drawing board. I kind of like that analogy because you do have a sick lake there. It's overly eutrophic, and it's been that way for a long, long time. And it's the way it is. I think we're running out of time because we could have you talk for another 3 hours. Honestly, this is a really interesting topic. John, do you want to close us out, John, just by any last words before we sign off?
Not really. I just echo your words. This was even better than I thought it would be when I first asked you to join us. Thank you very much for agreeing to participate here, and
Yeah, we'll have to have you back.
Oh,
Maybe
yeah,
next,
anytime.
maybe next summer.
A question, how did they fix Lake Erie? When you said anecdotally, in the 60s, I can tell you, we used to throw that muck at each other.
Yeah.
You know, I mean, it was thick, it was.
Well, federally, they banned
How
phosphates.
did they
in the-
clean it up?
They banned phosphates in the 60s from detergents. And so that helped a lot. And then the zebra
Oh,
mussels.
yes, but Toledo still has it.
Yeah, well, then the zebra mussels came in the 90s and started screwing everything up again.
Yeah.
So it was getting better, and then it's been getting worse. So yeah, and climate change and all these other factors that are stressing the system. So yeah, the detergent ban on phosphates was a huge component, really turned it around. That was basically called the dead lake, right? And they turned it around. It was good legislation, science-based, like yes. And then we still didn't quite get it right, but we learned, right? Now we know. So we're continuing to learn all the time, and we always make mistakes, and then the best thing you can do is learn. Yeah.
Right. Well, thank you once again for joining us. This has been really good.
Absolutely.
And we'll have it recorded. I'll try and figure out how to get it on our website so that, you know, other members can watch it who couldn't join us tonight. But thanks again so much, Dr. I'm going to call you Dr. Courtney, but I can call you your mother's saying, can't she say Wigdahl-Perry? Anyway, but thank you again. It's been wonderful. And I really want to audit one of your classes because it sounds fabulous.
I like talking to folks.
All
Yeah.
right, everybody. Thank you so much for joining us. We're still in the process of seeing if we can pull together
I think she found the new home problem here. She's currently in the process of moving, so she's in her new home, so she's probably in there.
And if we can, we'll let you take care. And yeah, I'm in the only room that's finished at this point. The rest of the place is a construction site. So anyway, thanks to everybody for joining us. And Dr. Courtney, thanks so much.
Thank
Take
you,
care.
John, for organizing.
Thank you.
Yes, thank
All
you
right,
very much.
yes, John. And John, if you wouldn't mind staying on, I got a question for you when everybody else signs off.
Okay, sounds good.
All right, take care,
Courtney,
everybody.
thank you, Courtney.
Yes, absolutely. I'll just, Andrew Hartley had some questions on some climate change, but he left and he asked for, but if you wouldn't mind, you could please ask him to email me directly and I can send him some.
Okay.
I just didn't, he said it right when I was starting. I didn't have a chance to reply before.
Right.
We started. So please just give him my information and I can reply to him directly. Just my email address is great.
Okay.
That right away.
That'll be fine.
Great.
Thanks
Thank you so much. Appreciate it.
Okay,
All right.
thanks, Courtney.
Thanks.
Bye, Courtney and Cindy. Mom, you did a great job.
They're managing the home front while I'm doing this. So all right. Bye-bye. Thank you.
Take care. Bye-bye. So, John, my question for you has to do, I was at Wegmans tonight, the one on Transit Road. Do you ever shop there?
Oh, yeah.
They don't recycle plastic bags.
Yes, they do.
They told me they don't. I was just there. I had plastic bags to recycle. And I said,
You're talking about the Transit Road East Amherst, right?
Wegmans, yeah, on Transit.
Yeah,
And
the
I went up
bin
to
for,
the customer service desk, and because I didn't see any bag recycling.
yeah, the bin for plastic bag recycling. Sometimes they have it in the bottle room, in addition to the one that is literally right next to the door on the left. As you're looking at the face front of the store, it's on the left door.
Left side, okay. I went in the right side, and
Yeah, there's nothing on the right side. All of the
okay.
The cardboard, the plastic bags, the miscellaneous bottles that you can't recycle. That's all closer to the bottle return area.
Okay, I'll check that next time if I'm out there, because and they didn't know. They said, "Oh no, we don't take plastic bags at this store." I'm like, "Huh?"
Yeah, they're required to do so, and they do so. for a while there, they lost the sign on the bin where it
Okay,
was like, what is this for? But
okay.
it is definitely there. They are definitely recycling. Matter of fact, in the customer service area for the first time, I actually saw actual recycle bins in their in the back on the back end there, so that
Yeah,
they can get rid of the paper and whatever miscellaneous, and so that was that was a new step. One of these days, I would like to contact there's every year for Earth Day, you get this email from Weichmann's talking about their sustainability initiatives.
yeah,
I want to reach out to the people at Weichmann's and see if we can actually get somebody
yeah,
to do that with
yeah.
us.
Talk to us about it. I mean,
I don't know about talking to us, but talking on at least on a higher level. Grab the Rochester people, grab the Syracuse people, and meet up in their Rochester offices or something like that, where
Yeah.
so that we can hear what they're doing. We can yell at them for shucking corn and putting it in a styrofoam container and in plastic wrap, stuff like that. But
yeah, yeah, exactly.
and then charging 3 times as much as it costs you to shuck yourself.
Right. Yeah, and it loses nutrients, by the way, when you take it out of the husk,
Yep.
because it's now exposed to light and oxygen. You're better off
Yep.
having it in the husk. There's no doubt about it.
No, it's not just that, moisture.
Moisture too, but yeah, I mean, most nutrients degrade with light and
Right.
oxygen and heat. Those are all bad for, for most vegetables and also fruits. I mean, there's a few exceptions, like bananas and tomatoes should not be refrigerated, but almost everything else, you're better off with it in a cool environment, if not refrigerated. So yeah, I agree. That would be interesting. And they're supposedly one of the more progressive companies around, even though they're anti-union.
They're anti-union, yeah.
Yeah, they're anti-union because I worked for them and they did an anti-union video as part of the orientation.
Really?
Yeah, Roger would not be happy. Don't say anything to Roger. But yeah, I remember that vividly, and I thought to myself, oh, this is not good. This is one thing I don't like about this company. They didn't have to show this video,
No.
But anyway, I digress. But I
No,
think
any company worth a grain of salt needs to show their employees for training the Krusty Krab training video.
oh, what's that? I haven't heard of
Season 1, season 2, season 3 was one of the earlier issues of your earlier series of SpongeBob.
Oh,
And they had a Krusty Krab training video that is absolutely beyond funny.
was it really?
If you should
on YouTube?
Google and see if it's on YouTube. It probably is, because like I say, it's so old that somebody's had to have put it online.
Okay, I'll
But
check it out.
The thing is, basically the punchline is, don't be a Squidward, because Squidward, they show SpongeBob and Squidward through the whole thing, and Squidward doesn't do well. He does the minimal possible, except for that that attempt at sounding like a clarinet, sort of like Colonel Klink's attempt at sounding like a violin player.
Right. Oh God. That okay.
Yeah, that's about the only thing that Squidward actually does. SpongeBob, on the other hand, is the beyond stellar employee.
Okay.
And the punchline all the way through, every time something happens, don't be a Squidward. It's like, it is well worth finding.
Okay. I will check it out. And what's the name of it again?
It's
The
a Krusty Krab training video
Krusty Krab training. Okay, I'll look it up.
with K's.
With K's, Krusty Krab. Okay, that's easy to remember. So,
Yeah.
John, thank you for arranging for
Oh,
Dr.
this,
Wigdahl-Perry. She was fabulous.
yeah. I mean, the only bummer I had is that the sustainability director for Chautauqua said that he would pop in, give a 5-minute spiel on
Yeah,
what they're doing in Chautauqua, but other than that,
didn't show, but
yeah.
you know,
But then again, yes,
yeah.
I'd like to know how to get more
Over
people.
13 people, and everybody seemed very interested.
Yeah,
So,
I'd like to know how to get more people though. It's not like I didn't reach out to every possible endeavor. I sent it to Brenda Young for the
posted it.
her environmental newsletter. I posted it on the Buffalo News. There's a Western New York gardening newsletter that comes out that she agreed to put it on that. Twitter, Facebook. We had it in the emails. We gave it enough time to start up.
You know
13
what?
people.
I mean, honestly, John, you did your best. And Caitlin put it out. She put it
Yeah.
out 2 or 3 weeks ago. So
Yeah.
people marked it on their calendars, put it into their computer and had it pop up as a reminder. I don't know. I don't know what else to do. And it makes me wonder. I mean, here it is, June 1st. It's not even, we're just starting the summer. No wonder Bob doesn't want to do anything in July and August. He says we won't get anybody there. I mean, we'll be lucky to get anybody later this month, to be honest with you. He wants to try and do another one this month. I'm like, really? I don't know. There's a part of me that says, if you get 12 people, you'll be lucky again. Maybe he has more people interested because on that energy committee, he's got like 25 people. And if the topic is transmission and upgrading the grid, those same 25 people will probably show up.
Yeah. Yeah.
But you-
The Sierra Club Waterkeepers. Let's see, the ACES group in East Aurora, which has a fairly decent following as well. And Andrew's group, he's with academic. He's one of the ecumenical groups.
Oh,
I think the United Church of Christ or something like that. I can't remember which one it is, but he's he's pretty well connected in that regard. So it went out to places that
yeah, I know. Now, as far as connecting people with Dr. Wigdahl-Perry, can we send out her email? She said like Andrew had questions. I didn't even notice the question. I was so
no, she could probably got a personal hang on that one because I looked and I didn't see any messages.
Yeah, I didn't see it either because I was checking questions. I didn't see it. So maybe I don't know if there's a way to send out her email to the people that were on this so that if they had follow-up questions, they could email her.
Yeah.
She wouldn't mind. Would you be able to do that? I don't even know how to go back and we've got this recorded.
Yeah,
but-
if you got the recording and you have, see, if you have the recording and you have the